Hi, and welcome back to Food Safety Office Hours. I'm Abby Snyder, an associate professor of microbial safety at Cornell University. I am co-hosting today's session with Wendy White, industry manager for food and beverage at the Georgia Manufacturing Extension Partnership. And today we're talking about process validation. So our speakers today include, of course, Wendy White, who works with manufacturers across the state of Georgia and beyond. And we are joined by Brian Farina, Senior Director of Technical Services at Dival Labs. And finally, we have with us today Kaitlyn Casulli, founder and consultant of Riskbusters LLC based in Athens, Georgia. So I'm going to start by asking each of these speakers to give us a brief presentation, and then we'll turn the recording off and move to Q&A. So I have a list of pre-submitted questions, but feel free to drop any questions you have in the chat at any time, and we'll take them at the end. And with that, I'm going to let Wendy kick us off.
Hey, thank you so much. All right. Can you see these slides?
Looks great.
Sorry about that. Wow. I wanted to thank Dr. Abby Snyder and Alex Salar from Cornell so much for putting together this fantastic webinar series, and especially to my fellow panelists who are really the experts in low moisture foods. So today I'm going to start us off with a broad overview of validation, and Brian and Kaitlyn are going to really add more depth on the practical application of validation and respect to the low moisture foods. So before I get started, a quick note on my department. The Georgia Manufacturing Extension Partnership is a public-private non-profit that's focused on helping manufacturers across the state. So we're part of a bigger MEP national network, and centers just like us are all in every single state, and we actually have one in Puerto Rico as well. They all offer training classes and subsidized consulting services. And the Georgia MEP is housed at Georgia Tech in its Extension Outreach Division, which is called EI Squared. So I wanted to start by defining a few terms that are often mixed up. And these are applicable to both FDA food safety plans with preventive controls and USDA HACCP plans. So when you're looking at these systems, validation is usually the thing that comes first. And it's really the evidence that proves that you've made good decisions in your hazard analysis and that the controls that you've selected are sufficient to control those hazards identified in those hazard analysis. Now, it's basically answering the question, will your food safety system work? Now, monitoring takes place every time that you run production, and it's the measurement or activities that are taking place to understand what is actually happening now. It answers the question, are we currently under control? Is our food safety system currently working? Now, verification also takes place on a continued basis, and it proves that you're doing what you said you're going to be doing in the food safety plan. And that you're monitoring and documenting your controls activities correctly. But today we're just going to talk about the first one, which is validation. So validation actually doesn't happen all that often. Typically, you only need to validate a piece of equipment or a control activity when something is introduced or changed. Now, this could be a new or changed finished product, raw material, piece of equipment, or a process, like if you add a new line. It could also happen if there's new food safety information, such as an emerging hazard or a recall that we've never seen before. Revalidation might also be triggered if a food safety plan reanalysis suggests a need for a change, or if there's a new regulation that indicates you need to revalidate. So when these things happen, you should start the validation process before the change is implemented or as soon as possible. And once that change takes place, you usually gather data for the first 90 days and analyze it to ensure that the change is sufficient to control the identified hazard. If the data doesn't support your decision, you might need to reevaluate your food safety plan or your HACCP plan. So, validation is usually composed of two different components. First, you gather your scientific or technical support documents. Now, these could be peer-reviewed articles in scientific journals, like the Journal of Food Protection. They could be predictive models to understand how pathogens will behave in certain foods under certain conditions. Other reliable sources of knowledge are university extension and valid professional organizational websites. And lastly, there are many USDA and FDA guidance documents which provide information to help you establish critical limits for your controls. Now, next, you'll need to collect some real-world data, which demonstrate that these controls are effective against your identified hazard and that your conditions are correct in your facility. So these might be in-house experiments, monitoring data from production, or challenge studies performed by a consultant or a laboratory. So what does validation look like exactly? My good friend, Dr. Brian Chavez from the University of Nebraska once told me that there's no one-size-fits-all approach to validation, which I know can be really frustrating when you're going about this. A good validation strategy usually combines several types of evidence and is tailored to your specific hazard, your product, equipment, and process. For example, let's say you're making a pickled food and you need control for Listeria monocytogenes. Your validation might include understanding the conditions that will stop Listeria from growing in peer-reviewed journal articles. Then using predictive microbiology models to estimate whether your pH and salt levels are sufficient to control that hazard. Next, you then conduct a challenge study to test your actual product formulation while testing for the acetic acid content and maybe the water activity over the full shelf life of that product. Now, that product might be purposely contaminated with a pathogen or an indicator organism. So during this time, during that shelf life, you can conduct periodic testing to understand how your controls, in the case of pickling, it would be the pH and the water activity, will affect that organism over time. And together, these pieces of evidence can help show that your process consistently keeps Listeria under control. An important note is that when conducting challenge studies that involve microorganisms, never, ever bring a pathogen into your facility. If you want to use a pathogen, let an outside laboratory take care of that for you. All right, so what needs to be validated? According to the FDA, you're required to validate process preventive controls in a FDA food safety plan. On the USDA side of things, they require validation to be done for all HACCP critical control points. Now, this includes ensuring that equipment responsible for those important controls, like a roaster for nuts, are tested to ensure that the equipment is capable of reaching that critical limit that you've determined, which will control the hazard. Technically, prerequisite controls don't have to be validated, but it's a really good idea to validate any control that's responsible for food safety. So you can see that allergen controls, because of from a governmental standpoint, don't have to be validated unless they're preventive controls or CCPs, but it's a good idea. Few final thoughts. You must document your validated activities and keep this data and all the supporting documentation, like your journal articles, the things that you've collected, for the life of your product. Depending on the nature of your product or process, you might need to bring in outside help. There are various trainings available. Dr. Casulli teaches a challenge study workshop each year for the International Food Association, the International Association of Food Protection. And remember that validation is the scientific backbone of a strong food safety plan. It turns assumptions into evidence and gives you the confidence that you need, that your control measures are really working. A well-executed validation program improves safety, supports compliance, protects your brand, and gives you a lot of peace of mind during those inspections and audits. By combining solid science, facility-specific data, and ongoing review, you can build validation strategies that are practical, effective, and compliant with current regulations. So now I'm going to turn it over to Brian from Diable to delve more into the topic of validation as it pertains to low moisture foods.
Right. Thank you so much, Wendy. Go ahead and make sure we're seeing the right stuff. All right. We all good as far as seeing what we need to see? All right. So what I kind of would like to talk about for this session is just some kind of fundamental concepts, some, what we call validation fundamentals or considerations, and then just do a brief overview of some of the major equipment types that we see a lot of when we do low moisture food thermal validations. So quick rundown, basically what I just said as far as what we're going to cover. And I want to kind of cover some key terms just to kind of set the stage a little bit. And really, it's just looking at the difference between in-lab studies and on-site studies. So an in-lab study is one that's performed not in the production facility. It's typically performed in somebody's microbiology lab. And it's done using lab equipment to effectively simulate the production process. One of the benefits of an in-lab study is that we can use actual pathogens when we're working in the lab, but there are some watch-outs, and there's kind of two big ones. One is the lab really able to adequately recreate the important aspects of the thermal process? And then on the other side of that, can the plant implement, monitor, verify the findings from the lab study? So in other words, does the study scale? So if we're validating, let's say, oil roasting of cashews, and we say that the cashews have to see 265 degrees Fahrenheit for 3 minutes, let's say, is the facility then able to ensure that every cashew they process through that oil roaster is going to see that time and temperature? And we'll talk a little bit about how maybe sometimes that's not the case. So the other type of study that we most typically see are on-site studies. So these are actually done on the processing line. And per Wendy's point, we never, ever, 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 ever bring pathogens into a food facility. We want to use a surrogate. We want to use an appropriately qualified surrogate, which is a bacterium typically that has equal or maybe somewhat higher thermal resistance than the pathogen we're concerned about. And so we can derive the kill that we would expect in the pathogen from the kill that we see in the surrogate. And that restriction about never bringing pathogens into a plant lab, that includes on-site labs. If there's a QA lab or a pilot plant within the facility, still do not want to bring pathogens there. We also don't want to look at avirulent strains of a pathogen or members of the same genus group, like using Listeria innocua or one of the other listerias in place of L. mono. It's going to mess up your environmental monitoring. Even if there's not a direct risk, it's not a good idea. If you're going on site, there needs to be a surrogate. Some of the watch outs there is that it does require line time. So we got to make sure that time is set aside for those studies. There's the question of what to be done with the product that is run alongside the study samples, because we have to run the unit at full capacity in order to effectively validate it. So what do you do with all the rest of the product that was being processed during that? And then what kind of follow-up cleaning and sanitation do we need to do after that validation to make sure that we're not, giving ourselves bad micro results or ATP results or, things like that. So getting now into the kind of four main topics or four main considerations that we look at in thermal validations. The first one I want to talk about is moisture and water activity, which is kind of the least obvious of the four, I think, and the one that we see either missed or not considered when we look at studies. So I wanted to hit that one up front. Functionally, at the level that the bacteria actually care about, it's really about water activity. And water activity, it's a technically it's a measure of a vapor pressure, the amount of water vapor that's coming off of the product as opposed to what pure water would be. Functionally, what we're talking about is how much free water do those microbes have available to use for their own metabolic processes. And it's a combination of how much water there is in the food to begin with, and then what kind of solutes are there and how much to bind up that water. So even though at a kind of bacterial eye view, that's what we really are interested in, a lot of facilities don't have water activity measurement capabilities, or they don't know what it is, or maybe they only do it on the finished product, not on their raw materials. So very often the conversation shifts to moisture rather than water activity, but the same kind of rules apply. So we kind of use moisture and water activity interchangeably. Moisture because functionally that's typically easier to work with, and talk about water activity because that's what matters to the organisms. And the reason why this really matters is that bacterial pathogens are typically more thermally resistant in a low moisture or water activity food matrix. And salmonella is the poster child for this. There's been lots of work done on salmonella thermal resistance at different water activities. This is just one. This is a snapshot of a publication where they use different sugar solutions to look at the thermal resistance of salmonella. Just a quick overview. Our highest water activity is 0.5. That's this red line. Our lowest is 0.18. That's the black line. And what they're looking at is the number of minutes it takes to show different levels of log reduction. So, at a 0.5 water activity, they saw almost 5, looks like about a 4.5 log reduction in 40 minutes. At 0.18, they took about 220 minutes, so that's like 5.5 times as long, and they didn't even see the same kill. This is probably like just shy of 4, or maybe 4, whereas the other one did much better. So absolutely, water activity matters, and it's going to affect how the organism, either the pathogen, if we're doing a lab study, the surrogate, if we're doing it on an on-site study, how it responds to the thermal process. Which means that any moisture that the group conducting the validation adds to the samples, maybe as part of the inoculation or how we prepare them, all of that extra moisture has to either be controlled, accounted for, or removed before the thermal trials. Because if you go into your thermal trials with samples that have a higher moisture water activity than what the normal product is, you're overestimating your thermal lethality. And that's cheating. You're getting more, you're being assigned more of a kill than you're potentially actually getting in the system. So there were some of the questions that we got in were about, what do we do in a process where we're effectively starting with a liquid product or a high moisture product, and then we're very rapidly drying that product. And so spray drying is a really good example of that. Dehydration, freeze drying are also good examples of that. And the thing about those is it is really hard to validate those processes successfully. Because what's happening is you're very quickly taking the product from a higher moisture state, water activity state, to a much lower one. So you're kind of on the fly, very rapidly changing the thermal resistance of the organisms and making it harder to get the kill. It's kind of like in the movies when they're in some kind of facility and someone hits an alarm and the big security doors and windows come slamming down and you have to try to jump through it before that happens. That's kind of what happens with the bacterial resistance. Not really, but kind of. It's a fun analogy. When you just change the moisture so quickly, the best way and the most effective way to validate any sort of rapid drying type process is to get your kill step in, or render the product ready to eat before you start that drying process, while it's still a liquid, while it's still high moisture. That's where your success is going to come in the product then. And then do whatever drying you have to do, following that, and making sure that your GMPs, your sanitation, et cetera, are in place, so you're not reintroducing anything. Temperature is another obvious important consideration, but what we want to talk about here is that there's very rarely just one temperature that we're looking at. So, the obvious one is that the set temperature or the target temperature for the process. But there are others. What temperature does the product start at before it goes into this thermal step? And how does that change? What are the actual temperatures that the unit is delivering versus what it's set to do? And a really good way to get a handle on this, if the process can support it, is to do temperature mapping. Temperature mapping is an incredibly valuable tool. And what we mean when we say temperature mapping is to use independent thermocouples. So not the ones that are connected to your recorder or your recording chart or anything like that. These are separate. These are independent, and they're distributed throughout the product. So they're actually able to record the times and temperatures at product level. And so here's some examples. These are graphs that we actually generated as part of client studies. The one on the left is actually a batch process, I believe it was an oil roast process where product was dropped in, and you see how the temperature drops as a result of that. The second one is a continuous process that has two different heating zones. And what we notice, it's kind of hard to see based on the size of this, is that each one of these increments on the y-axis is 5 degrees. We've got about a 40-degree difference on this, the left example, the batch process. We got about a 30-degree difference in zone 1, this first zone, on this continuous process. And without that mapping, you would never know that that was there. Next, we want to talk about time. In low moisture foods, there is no such thing as killing based on temperature alone. That's pretty exclusive to high moisture products, meats, things like that. There's no instantaneous kill in a low moisture product. There's always a time and a temperature. And it's important to account for all of the different times that are potentially in play. So, we've got the time spent in the processing step. But what about, warm up time? What about cooling time? What about, any kind of hot holding after the fact, like retention bins or, whatever? Those can all factor in. And so if you're counting on those as part of your kill stat, those have to be considered as part of the study. And a big one that I see, especially in, places where you've got, drum dryers or things like that, is, do you have a way to make sure that all the product you're processing is going through the kill step at that same minimum time? We've seen big drums where, the product, there's no fins to kind of keep things in line, and the product just kind of slowly kind of makes its way through the drum. Maybe there's an incline or something to get it out. And there's always, you can see, little bits of product that kind of dance across the top of everything else, and they come out of the process quicker. And so how do we account for that? And then the fourth one, the last of our considerations, is product flow through. So how much product is being processed at a particular time in the unit? So batch process, it's pretty easy. It's the size of the batches that you're running. In a continuous process, that can be measured with bed depth, can be measured with flow rate. But we want to make sure that whatever type of process we're validating, we want that system run at full or maximum capacity. If we run at with a smaller batch size, or even wouldn't do this, it's kind of crazy, run samples through the system without accompanying product with it, you're not going to get an accurate representation. And you need to think about, how are we going to get the samples in? Where are we going to put the samples in? Are we going to contain them in something? And what is that containment affect? Does it affect heat penetration? Does it affect how those samples move through the process? Are they moving at the same rate as everything else? And again, circling back to what we said about lab studies are typically done on very small portions of product. So is that going to scale into a production level process? So Kind of going into the second portion of the talk, we're talking about some of the different units that we very commonly see when we do thermal validations. First one is a continuous either hot air oven or roaster. We see this a lot with nuts and seeds, cocoa. We see this a lot with baked products that are on a conveyor oven system. As far as the things we want to make sure we're accounting for in these types of validations are things like fan settings, damper settings, what the airflow is for each zone, what temperature is the product when we put it in. Are we actively cooling the product, or are we letting it kind of cool on its own in ambient air? Bed depth, flow rate, that's that fourth principle we talked about. Belt speed, time in unit, that's the third principle we talked about. And then the temperatures. What are we setting for the different zones in the unit? And then what are we actually seeing in those zones during the validation trials? Hot air, though, is my least favorite mechanism to try to validate, because hot air is very squirrely and it can be really challenging to get hot air to go where we want it to go consistently. So there's oftentimes some kind of extra factors or extra work that has to be done to make sure you're prepared for a validation. Air balancing is a big one, making sure that the hot air is going where we want it to go and isn't going off in a different direction or out the infeed or out through the cooling zones. There are baffles that control the direction of the air, and if they're not set right, you can go off balance. Fan speeds, we've seen validations where the fan speed in the first cooling zone was so high, so vigorous, that I was pulling hot air out of the last heating zone. And then belts, that hot air has to pass through the belt. So if we change the belt or the size of the perforations on the belt, or if we're working with a dirty belt where there's soil that's clogging up some of those openings, that all factors in to the kill that we'll get. The other big consideration is what we call leading edge. So when you're looking at a process where the bed is fully packed, fully filled with product. So we're not talking about, bake processes or things where you've got significant sections of open belt in between where the product is. If the belt is entirely full, their concern is what we call leading edge. And so if we think about these red arrows as the hot air, when you've got that very front edge of product going through, it can be easier for the hot air to go out and around it rather than to go through the belt. Through the product bed, because it has to once the bed's full. So we want to make sure that we're accounting for this, because this area very often gets under processed. Second type of units are kind of batch or rotary hot air units, and a little simpler. There's usually some sort of burner setting. Again, the product of the temperature going in is really important. Batch size, dwell time, the temperature of the unit before product's introduced, what the target temperature we're processing to is. That's usually at the end of the process. And then are we doing any kind of hold once we hit that target, or are we discharging? We hit a temperature and we discharge versus hitting our target, holding it for so many minutes, and then we discharge. Watch outs for these systems is that this is where we see a lot of kind of older units that don't have great monitoring, may not even have a set temperature. They just have something, gas turned on to 75% or something like that. It can be difficult, especially with older units, to track the temperature of the actual product. And this is where we often see what we call artisanal processes. And artisanal in validations is not a good thing. Artisanal means, we kind of process till it looks right, or till the moisture is where we want it to be, rather than having, strict controlled times temperatures that we would typically look for. Now we have continuous oil systems. So similar to continuous air, and that product is continually flowing through, but because we're using oil as our heating mechanism, typically more reliable, more constant. Not bulletproof though, because we still do see issues with hot oil systems. Same basic parameters, time, time, set temp, actual temp, product flow through, belt speed, nothing too crazy there. Batch oil fryers or roasters are kind of the other type of unit that we see a lot of. And this is where, you've just got a bath of hot oil, hot water. They also use hot water a lot for like pecan pasteurizing and things like that. Same idea. And then we drop the product in and then we hold it for a certain amount of time. So, burner settings, if we don't have a direct measurement of temperature, batch size, in feed, again, always important. If you're going into a thermal process with product at 50 degrees Fahrenheit in February and 80 degrees Fahrenheit in August, that can be very different on the load that it puts on the unit. And this is for any of the types we've talked about, not just this one. So that's an important thing to think about and to consider. And it's the batch oil systems. This was the one where we saw that really wacky 40-degree difference temperature mapping. Because as soon as you drop the product into that oil, its temperature is going to drop. Does it come back, or does it come back to? Are we effectively getting the same time and temperature to every piece of product everywhere in that basket? All important things to look at. And then finally, we have extruders, where we take some kind of a base flour or meal or something, and then we turn it into typically some sort of puffed or expanded type product. These are a little different. Some of the main themes are definitely still there. With an extruder, we added a certain type of die that we might be using, which can change how the product passes through, the friction that we generate, the heat that gets generated through that. It's really important to know what the temperature is at the die head itself or at the product coming right off the die head. That's usually the temperature. One of those two is what we really focus on. Your screw RPM is going to give you your time. We see our throughput feed rate. Lots of extrusion processes either add water or steam as a preconditioning step or as part of the actual extrusion process itself. We want to make sure we're accounting for that. And then there are some facilities that use the moisture of that extruded product or the water activity as an indicator of whether they've adequately processed the product. So they have it has to be below X for us to know that we've properly processed the product. Not everybody, but in some facilities, that's an important thing to work into the study because they use that data. All right, that was super quick, and thanks everyone for hanging in there. I will pass over to Kaitlyn. Thanks so much, guys.
All right, that teed me off pretty well for my presentation. So I'm just gonna get things straightened out here. Zoom back. Hope that one is correct. All right, so that should be showing my presentation right now. And all right, cool. So I am going to present on some data that I collected during my PhD. So I got my, actually, let me just go ahead and flip to that next slide. I got my master's and PhD from Michigan State, and while I was there, my main focus was on low moisture foods. So for my master's, I worked on developing a model for salmonella inactivation in pistachios, and after I completed that, I did a little bit of consulting. I worked on a project for risk assessment in peanuts, and I was inspired to dig deeper into peanuts for my PhD project. So after I finished that project up, worked with a company that was doing dry roasting, started my PhD, and was able to work with a roaster in Georgia, no less, where I actually ended up after my PhD. And so that is what I'm going to present today, is the data from that roaster. So I did put a quick intro slide, but I think both Wendy and Brian took care of this for me. But just kind of thinking about the inspiration for this was FSMA and the preventive controls rule. So with FSMA, we're trying to prevent foodborne illness rather than react to outbreaks. So once we identify hazards such as salmonella, usually we're looking at thermal means for this. And a lot of times with the low moisture foods, especially in the nut and seed industry, we have these technologies in place that are meeting a quality standard, legacy technology we call them. Some of the equipment is kind of old, some of it can't really be modified, so you have to really get creative about how you're going to validate these things. And the goal is, not to have to replace the equipment, but to just adjust the settings, and not, having the burden of having to spend $1 million on a dry roaster if you don't need to. So Brian already covered, but dry roaster, this is, I believe this is actually the one that I used for the work that I did. So this is a Buehler dry roaster, and we have the infeed or the hopper where the peanuts are loaded in onto that belt, and then dropped into the hopper, usually loaded from what we call the super sacks, giant sacks of peanuts that come from a warehouse, maybe temperature controlled, maybe not temperature controlled. So we'll get into some of the implications that I noticed from this work. There's the preheating, which will just kind of get you started. And then there are heating zones. The one I looked at, the roaster I used, had two larger zones, and then they were broken up into smaller subzones. So each zone had if you look at this, I'm trying to see if I can find a pointer here. No. But anyway, so if you look at each of the doors, 2 of these doors corresponds to a zone in the roaster. So a preheating zone, a heating zone, and then cooling zones. And then once the peanuts travel through that roaster on the belt, that's inside, it's just one single belt going from front to back. They get discharged, and then this little hopper will pull them out and send them off to packaging or post-processing or whatever is needed. So what I have been doing for the last several years is really getting into the science of the interactions. And this next slide is going to describe a bit about the interactions between process, pathogen, and product. So if we think about a single peanut being like a solar system or even a planet in the solar system, we can actually translate that to, say, a lot of peanuts. So a lot of peanuts is the Milky Way. Instead of just looking at a single peanut like I was doing for my master's research, I wanted to get into this idea of what does it look like when we think about a lot of peanuts inside the system? And what happens if those peanuts have a pathogen on them? Well, we have to think about process characteristics. So some of these factors could include the temperature, the humidity, the depth of the bed of peanuts on the belt, the air velocity of the air that's moving through the peanuts to heat them, and then the direction of the air. In the diagram that Brian showed, there were zones that had the air going up, and then there were zones that had air going down. And that's really meant to even out the differences that could exist on the bed of peanuts based on how deep they are. So if you think about air coming up, that bottom layer of peanuts is going to be a lot hotter than the ones on top because the air is hitting them faster, transferring the energy to the peanuts, and then there's less energy for the peanuts on top. And then other product characteristics. So the moisture in this case is decreasing throughout the process. Moisture, also water activity, is decreasing. The temperature is increasing. The composition of the peanuts could be variable based on the time of year that they're harvested, or even just the general climate of the area at the time, the size of the peanuts. So if we think about pouring a bunch of rocks into a glass versus pouring sand into a glass, the airflow is going to be different between those two things. So even if, water is also a fluid like air is, so if you pour water on top of those rocks, it's going to go down to the bottom of the glass very quickly. But if you pour water on top of sand, it's going to take a while to really penetrate through that sand. So size of these products actually makes a pretty, could make a significant difference. And then the thermal properties, how quickly are these products heating up? So if we think about where salmonella is, it's on the surface of these products. So we're really interested in kind of the effect of the rapid heat transfer from the environment to the surface of that peanut, and that will really drive the inactivation. So I did a lot of this. I did kind of the fundamental lab stuff during my PhD, and I had this massive data set, and I didn't quite know what to do with it. That was, I'll describe how I got that data set. But the overall goal and objectives for this portion of the project, which I completed after my PhD while I was still with University of Georgia, so the overall goal is to develop an enhanced understanding of pathogen reduction achieved during roasting of low moisture foods by incorporating these product and process factors into the modeling. So for this particular portion, I had 1 year of production records of a dry roaster. And I'll show you exactly how I got those records. They're temperature records. And then with those temperature records, apply a model to temperature the data that was collected in the peanut roaster, and then use the lab scale model that I developed for the Enterococcus faecium kinetics on peanuts to actually figure out what is this dry roaster doing and what factors are influencing what's happening in that roaster. So this is a top-down view of the roaster. Again, intake and discharge, intake being on your left, discharge being on your right. I mistakenly said I had, no, I do have 2 zones. They're just, I mislabeled these. So the 2 boxes on the left, zone 1 and zone 2, are actually zone 1, and then the next 2 are actually zone 2. Just the way that they're divided up, they have doors. So I just labeled them based on the doors. This was a 4-door roaster, 2 zones. Inside each of these zones, I had Bueller, the company I was working with. And I did pay them to do this. They didn't do this for free. So so I had them put bars across the width of the roaster at 6 different points. So I had them put row 1, row 2, so 2 rows in each of these subzones for the last 3 sections. So behind each door, there were 16 sensors. I didn't do anything on the leftmost zone because that wasn't really going to be contributing to the lethality. So based on some of the lab scale data that I had, I figured out that the temperature really became relevant only once you got to that second door in that zone. So there was no need to really fill it with sensors. Sensors are expensive. So I decided to really focus on the most relevant parts of this roaster. So in total, we had 48 sensors, and then kind of visualizing how it was subdivided. These 48 zones, if you're a peanut, imagine you're walking down one of these, one of the 8 lanes. You're walking through this roaster, and once you touch the edge of row 1, your temperature is being updated every 10 seconds by that one fixed sensor until you hop over to the next part of that zone. So each of these blocks, the temperature would update that little section of peanuts. We call it plug flow. So the section of peanuts that's represented by 10 seconds on that belt gets updated continuously by that sensor until it moves into the next section. Then it's going to get updated by that sensor, and so on and so forth, until it moves through all 6 of these sensors, and then you can make that temperature profile. So to code this, I took all of the data. So they sent me 12 data files representing a month of production data. So all of the temperature data from each of these sensors over that month period, and then some other data such as, you know, the timestamp, the date stamp, the bed depth was measured, and then other kind of roaster parameters, you know, the airflow, the damper opening, whatnot. So I received all of that data, and it was, you know, not including thinking about how many columns, but there were over 10 million rows in once I combined all of this data into a single kind of file or data structure in R. Talked about that, combined everything. So each month I just stacked them one on top of the other. I divided the timestamps. So the timestamps came in a very standard reading, where they give you the year, the month, the day, the hour, the minute, the second. So I used that timestamp to figure out what was the residence time between the readings. So I could figure out how to calculate that cumulative lethality, which is based on the residence time. I extracted all of the temperature data when the roaster was operational. And the way that I did this was, it's not operational until you have peanuts in that roaster. So once the bed depth was over 3 centimeters, I extracted the clusters of data when it was operational. I then created the individual temperature profiles for these peanut plugs that I was talking about. So it's not enough to just say, okay, we have all the data points. We actually need to follow these peanuts through the roaster. And that's where I was doing the manipulation of, okay, you're traveling through that first little section, update every 10 seconds with the temperature from that section, and then jump to the next section all the way down the line. Ended up with a little over 3 million unique temperature profiles. So that includes all the profiles for each of the 8 lanes. So each lane had its unique temperature profile from those peanuts, from that coding where I was calculating those profiles. And then with these profiles, I used the model that I developed during my PhD for Enterococcus faecium in peanuts. And that model looked a little bit like this. Actually, no, it looked exactly like this, where the log of the survivors was determined with a log linear model combined with the Bigelow type model for figuring out how the inactivation changes based on the input temperature. So if you integrate that across the time, the residence time that it's in the roaster, you can figure out how many log reductions you achieve based on that model. So this is just a representation of the temperature data, just the raw temperature data in degrees Celsius. So ranging from about 140 to 160 on the x-axis here on the bottom. And I apologize, it's a little difficult to figure out how to show all of these data points in a nice cohesive way. But what we have here is 1 through 6 is representative of moving from intake to discharge. So interestingly, or maybe not interestingly, the temperature starts lower from being close to the intake, and then it gets higher at the discharge. I think the airflow was inverted around number 3 here. So the third column from the left is where the airflow got inverted. So that was just kind of observing the patterns here. And then along the right side, the numbers 1 through 12 represent the month of the year, where 1 is January and 12 is December. So one of the interesting things I wanted to point out is anyone who's familiar with Georgia climate knows that April is incredibly variable in terms of temperatures. You're kind of partway somewhere between winter and spring and sometimes summer. So the temperatures, they just fluctuate pretty wildly. And we can observe that based on the data. So these peanuts were coming in with the super sacks. Their initial temperature wasn't being controlled, so they're coming in and it's extremely variable. On the other hand, if we look at months of January and August, so number 1 and number 8 boxed in those blue boxes, January tended to run a bit colder in terms of the product coming in versus August, and we'll be able to see that a little bit easier on some of the next graphs that I'm going to show. This graph is similar to the last one, but so the months of the year are represented on the gray boxes at the top. So 1 through 4 is January through April. 5 through 8 is May through August, and then on the bottom we have September through December. Again, plot number 4, actually I should explain, sensor 1 through 8 is representing each of the lanes. So if you're looking at the roaster from the intake side, the left sensor 1 is the left side of the roaster, and the right sensor 8 is lane 8 on the right side of the roaster. So this is showing that we have a lot of variability between that left and right side. So when Brian was talking about the balancing of that roaster, balancing the airflow, it could be that this roaster is having a little bit of trouble with that based on these plots. And then we can also look at some of those hotter months and see that, crap, I should have mentioned, sorry, I'm rushing a little bit. But the y-axis, the values you're seeing are actually the cumulative lethality calculated by this model. So each of those profiles for that lane was represented by a single number for the cumulative lethality, and that's what the output is. So if you have a bigger, taller bar that's up higher, it just means there was more lethality. So more lethality from left to right, and more lethality during these warmer months when the product is coming in warmer, but also a good bit more variability. So it comes, trade-off, things come hand in hand. So it's definitely something to examine further, just how variable the system is. And this next plot actually is one of my favorite because this will tell you you're kind of looking again top down, left to right. Sensor 1 through sensor 8 is the left side of the roaster to the right side of the roaster. And now we have data that I labeled properly from January through December. And this is a heat map, so you can actually see visually, wow, July, kind of hot in Georgia. We're getting there right now. It's starting to warm up here in June. But that hot month has a lot more lethality than some of the colder months. So really what I got from this is, just balancing the value of making sure we're understanding what is influencing that left side of the roaster being a lot hotter than the right side in a lot of instances. And then figuring out if you're not going to climate control your incoming product, what would be the optimal time to come out and do a validation? Of course, sometimes if you open up your plant in July, you may not have the time to be able to sit and wait around until February to validate that worst case scenario where you're seeing the lowest inactivation overall in that system. The other thing I want to point out is the variability. So this is plus or minus a standard deviation for each of these. I've not handled very large data sets like this previously, so I don't know whether it's a me problem or a math problem, But I am very interested in working with someone who's very good at statistics, so I can figure out, what are some of the best ways to represent these data and how can we make it meaningful for the industry. So I don't really have a conclusion slide. That's my conclusions. I'd love to do further work on this, but I do want to acknowledge my PhD advisors, Kirk Dolan from Michigan State, and then Don Schaffner from Rutgers, and then Bueller for being generous enough to allow me to pester them into collecting this data. And with that, I'm concluding my presentation. Happy to assist my co-presenter, co-panelist in answering questions.
Great. Thanks everyone for the presentations. We are going to